सर तुमचा ऑडिओ म्यूट आहे.
Okay.
या गुड मॉर्निंग.
Good morning.
सर पीपीटी शेअर करून बघायचं आहे, चेक करायचं आहे.
PPT तो है. It is there with me.
ओके. स्लाइड शेअर करून एकदा चेक करायचंय ओपन ऑपरेट होते का?
Can you see that?
No sir. No sir.
Just 1 second. I will just 1 second. Screen share. So are you able to see it?
No sir, not as yet.
Not as
Let's wait, maybe.
Yeah, it takes some time maybe.
Okay.
No. No, shame.
No, not yet.
Not yet. Okay. Here,
If you-
optimize the video.
So if you keep the PPT open, it becomes easy for sharing.
Okay.
Just open and minimize that PPT which you are presenting.
Yeah, that only I am doing, minimizing it. So share it. Okay. Share sound. Okay. Just 1 second.
Yes, sir.
Screen. ये फाइल शेयर करनी. हाँ.
Yes, sir. Now
Yes,
it is.
yes, sir. Just please make it as slideshow. So yes.
Yeah.
Yeah, that's fine, sir. Okay.
Yeah.
So you're requested to stop the sharing for now, please.
Stop sharing.
Yes, for now, sir. Then it can be reshared after some time.
Okay. For some time, stop sharing.
Okay.
Right now, for some time, but
Okay.
later on again, it can be shared.
So next time just you have to start
Okay.
and press this.
Okay.
Thank you, sir.
Okay. Yeah.
Yes, sir. Can we start?
Yeah.
Are we live, Shivani madam?
Yes, yes, sir. We are live. We are live.
Yeah. Sir, should we start?
Yeah.
Okay. Shivani madam?
Yes.
Yeah. Balika sir?
Yes, sir. So again, after that short break, I welcome all the participants and today's speaker, Dr. Dhiman Sarkar, for this second session on the day 3 of the FDP. Let me introduce you. Let me introduce Dr. Dhiman Sarkar to you. Dr. Dhiman Sarkar is a person with a research management skills, and he brings in about 35 plus years of experience, which encompasses the laboratory level researcher to an expert mentor, and he has an experience with the industry, with the academia, and also he has a experience of creating technology startup company as well. So that's a entire span has been covered by him. He's an expert at looking for growth opportunities in healthcare sector with advising research projects in troubleshooting and management. Looking at his professional journey, currently since March 2018, he is a chief scientist at NCL and is mentoring PhD and postdoc students. He is working on target identification of lead compounds, also involved in resuscitation of latent tuberculosis bacilli. Also, since January 2015, he has started his own startup, Anveshan SciTech Private Limited. He is a founder director of the company. And the company is involved in developing a novel diagnostic kit for the detection of tuberculosis by using sputum samples. He has received funding for the proof of concept study from BIRAC and DBT also. Then he has been principal scientist and senior principal scientist and principal scientist between 2007 to 2012, during which he has guided the PhD students. Then he has started 2 major drug discovery projects which were funded by CSIR, and he has developed new dormancy models for tuberculosis. Also HTS, high throughput screening against different tuberculosis models and cell lines. He has also completed a project on developing botanicals in skincare, which was funded by Procter & Gamble USA. He has standardized protocols of in vitro dormancy models of tuberculosis and anti-tubercular screening of synthetic library. Earlier to this, from 2002 to 2007, he has been a senior scientist with CSIR, that is the CSIR National Chemical Laboratory, and he has created a new laboratory for tuberculosis research, started industrial projects as mentioned earlier by Procter & Gamble, and he started mentoring the students for completing their thesis. Earlier to this, from 1996 to 2002, he has been associated with AstraZeneca R&D at Bangalore, where he was a research scientist, and he was characterizing enzyme mechanism of anti-malarial drug target, and he closed a major anti-malarial project by identifying defects in the screening assays. He developed STS assay protocol for anti-malarial target for screening of about 1 million compounds. He has also developed assays during this tenure for MTB cell wall enzymes. Then earlier to that, during 1994 to 1996, he has been a research associate with CSIR, Indian Institute of Chemical Biology, Kolkata. He has studied the role of calcium ions on morphogenetic transformation of Leishmania donovani, and there he completed the thesis on biochemical studies on some aspects of host-parasite interaction of Leishmania donovani under guidance of Professor Amarnath Bhaduri. Then earlier to that, he has been between 1989 to 91, he has been a junior research fellow and senior research fellow at CSIR laboratory, Calcutta. And he has standardized the growth kinetics of Leishmania donovani and standardized different ways of infecting mouse for developing Leishmania infection. He has also isolated and purified LD amastigotes. So with that journey and the skills like R&D project management, drug discovery programs, then scientific writing, research training and guidance, research troubleshooting, entrepreneurship expertise. So he's a formidable speaker for the theme that we have for this FDP. His accomplishments are 12 PhD students, and still some of them are completing their work. Development of drug discovery screening laboratory with biosafety level 3 facility, publishing 145 scientific papers in the peer-reviewed journals, and he has filed 45 patent applications. So with that introduction, I would request Dr. Sarkar to start his presentation and guide all of us with his expertise about starting a startup company. Thank you, sir. Welcome.
Thank you, Doctor, for your nice presentation. Today, in this occasion, particularly related to innovation, startup, and this innovation management, and I have got actually a very nice theme on which I can speak something, but not a very elaborate presentation. So here I am going to mainly share my experience related to my innovation journey. So can you see the presentation?
Yes, sir. Yes, sir.
Okay.
Perfect.
Okay.
Yeah.
So today I am going to talk about my experience as scientist entrepreneur. Basically, company we made, the name of that company is Rationale Sci-Tech Private Limited, and it is basically a technology startup, and main target of developing medical biotechnology product, particularly the TB diagnostic kit. Apart from that, we have, as you know, that we have done some other works also. But today I will try to focus on the technology that we were trying to develop. So you can assume that because I'm a scientist at CSIR NCL, so Rationale Sci-Tech has got the technology from CSIR NCL. So as a result, Rationale Sci-Tech can be called as a spin-off company from CSIR NCL. So, and that is based in the venture center with a lab to market program. So our motto was basically to develop, manufacture, and market innovative products and beyond. Before going into the company and further details, I want to share that how we got that idea that the company has to be made. One day we were discussing actually with my old classmates, and they are already established and they are running some company like technology startup company, and they are looking for funds. And I told them that it is possible that if you help me in acquiring some data, the clinical data, then probably I will be able to help you, because I have some small project related to that at NCL actually. So I asked them to test the sputum sample by using certain reagents and test the presence of mycobacteria. I'm saying that this particular test can detect a metabolite biomarker. So I am hoping that if I get the positive result from that test, then it will indicate the positive TB. And if it is not, then it will be TB negative. And you can see that we had done on around testing of around 102 samples at that time. And here you can see that total positives were 68, whereas by microscopic technique that is used as a regular standard technique in all the TB detection laboratories, that 37. So this test was done by the hospital. So the test was done by the hospital, that smear microscopy test. And our people went there and used that same sample for testing by using our methodology. And that was the kind of difference. So what happened is that we were very surprised to see that kind of difference. And frankly speaking, I was having a mind that we will try to find out if it is possible. We will try to find out it as a methodology, new methodology, because we are getting. But frankly speaking, I didn't have a detailed idea about the methodology of analysis and other things. So after that, what we did is that we went for patent filings from NCL. So the patent applications were sent to PCT, then US, then Russia, South Africa, India as well, and European Union. So definitely some kind of thinking was there. I will come to this particular aspect later on with more insight. So if we so immediately what happens is that the thinking goes to that what kind of market for TB diagnostic kit could be available, or that what is the size of that market. So TB diagnostic market in India is worth around 550 million. So that means the sales goes to that level every year, and with a certain growth rate. So India's size is around 25% of the global requirement. And you can again divide this market into established market economies and outside of established market economy. Established market economy means that the developed countries where actually this kind of TB testing is not carried out. The occurrence of TB cases are very low there. Of course, there the dynamics are also very different. Means that the latent tuberculosis is more prevalent there, and active TB dominates in poor countries like India or third world countries, you can say. So WHO report at that time said that around 60% of TB cases diagnosed in LIC and 66% globally. So that means around 40% cases remain as unknown, or they are not detected. And the test, that average price for that has to be paid is around approximately 1,700 rupees or $30. that is basically the cost of TB diagnostic test. So it is actually a huge amount that has to be paid. And I can tell you that this 1700 is not just that amount that has to be paid. A patient has to go to different places and remain confused that whether he or she has TB or not, and before confirmation, they have to test 4 or 5 times to different places finally. So around 5,000 to 6,000 rupees has to be paid before coming to a kind of conclusion. So actually I cannot see this on. Okay. So the what is the need? Then we try to think of, and what is the novelty? Novelty means that we have filed for patent, and if that is accepted or granted, then we know that we for sure the novelty is there. And regarding the need is that there is no POC method, that means point of care method available for TB diagnosis. So there is definitely a huge need for such kind of product. And all the recently developed methodology, they are heavily biased on NAT, that means nucleic acid based test, which require skilled labor, sophisticated instruments and laboratory settings. The average cost, again, that is very high. So it will be very difficult for patients of low income group. So we decided to go for starting a venture where we can take the idea through development of that research into product and to finally to the market. So in that company, the basic participation was from my side, and then Manikankana Sarkar, and from Entrepreneurship Development Center, Dr. Lele. And along with us, the company was associated with Baheti and Somani for financial audit and related things, and for any legal kind of help, Lakshmi Kumaran and Sridharan from Bangalore. And we recruited a project assistant also. Actually, before that, during our project, 2-3 project assistants were also recruited. So our business model, we decided at that time to use our technology as a smear replacement technology, and that will be used in the early diagnosis of TB and treatment response. I have already discussed about the market size. So overall market size is valued at US $2.2 billion in 2016, and it is expected to reach to $3.3 billion by 2025. So the problem statement, we can say in a different way, that there is a huge gap in TB diagnosis and treatment. The current WHO endorsed TB diagnostic tools like microscopy, culture, and molecular methods like Gene Expert, CVNet. They are capable of detecting replicating bacteria in clinical samples. But there is no test available to detect those who are not replicating. As we know now very well that TB can nicely reside for years in our body without replication. So that is called, at some stage of disease, called latent tuberculosis. Someone says that VVNC, viable but non-cultivable. Someone says that it is dormant, it is non-replicating just. So whatever may be, you can say, but there are differences among those populations also. I am not going into that detail today. So our product is basically then a metabolite biomarker detector based novel TB diagnostic kit that can detect MTB, which is missed out by smear microscopy, which is used as a basic methodology for the detection of tuberculosis in most of the laboratories in India. Our test will be very specific to MTB in sputum samples, if we use sputum samples, and it will be rapid, easy, instrument-free, and low cost. So within $1 or $2, that means within 150 rupees, you will be able to get your test done. So now again, I'm trying to emphasize that what happens today, still today, and what they cannot do. That is AFB, that means microscopy, culture, and Gene Expert, nucleic acid based methodology. They detect only the actively growing bacteria. So when we will see that actively growing bacteria, because we will be able to see them only when the patient is showing all the symptoms of the disease, or they have grown up in such a way, this bacteria in the body, that patient has gone to almost in the terminal stage. And if it is in the early stage, or the bacteria itself is trying to survive very hard in our body, at that time we will not be able to catch them or identify them. And if there is no methodology to detect this non-growing bacteria, then also even after that, WHO report says that 40% of active TB cases are not detected. But to me, the question is that how you have been able to estimate that 40%. It could be 140% also. So nobody knows. So probably this is the reason why we don't see any major impact on TB eradication. And this non-growing bacteria, after some time, they are not killed by the current medicine, neither they will die also, and through our immune system. And after some time, looking at the situation, a favorable situation, these bacteria will come out and will make active TB. So those bacteria which are not detected by mainly the test carried out, that microscopy test, we can say them as missed out cases. So the rationale for our this new diagnostic test is that MTB can utilize its alternate respiratory substrate or some substrate metabolite related to hypoxia and or dormancy, and that can be detected as a biomarker. So the test should be very easy. So in that way, our test is very novel, and it can detect the dormant or non-replicating MTB, which actually is responsible for the lengthy treatment regime and treatment failure also. So this test ideally can detect TB in the early stage of the disease, and thus greatly can influence the decline in TB transmission, prevent new LTBI cases. LTBI means latent tubercular bacilli infection cases, and thus potentially save lives. So now, if I come back to the ordination on how they perform within these days and when they started. So if I talk about its journey, then it is established as a registered company on 13th January 2015. Then we submitted the project to BIRAC through IKP Hyderabad, and then we received the grant also in June 2015. So then the process started. We signed MOU with EDC for tech transfer in June 2015. So then we received fund from BIRAC and established the company setup at Venture Center in July 2015. The project was completed in March 2017. So within this time, we can say in terms of the progress towards the positive direction is that we developed the prototype of the TB diagnostic kit. Now, within this, what kind of data we acquired, we will give a little bit about that, some little bit data we will try to provide, that what was basically the theme of the work that I will discuss. But before that, I'm showing here how it looks like. This is the test that is carried out and how it looks like. So this is a positive test result, and this is negative. So here, because it is negative, there is no color. So it is TB negative sample. And here the color is here, so it will be TB positive. So from here, what we will know is that the presence of tuberculosis, the presence of missed out bacilli, probably more so, and severity of the disease also. So maybe I can elaborate it with more detail on the prototype. So this is basically a prototype kit. Here you can see that sputum sample cup, sputum sample collection cup that is provided, and two small sized bottles are given. In these bottles, two reagents will be provided, reagent A and B. And if you see here, that sputum will be collected first in this collection cup, and then you have to add reagent A in the sputum and mix it well and keep it at room temperature for 10 minutes. And then you add the reagent B to, then you will see that immediately, if it is TB positive, color will be developed. So the color intensity is going to depend on the number of bacilli. If the number, or in other way, severity of the disease. If the color is more intense, then it is more severe. And obviously I said that color is not there, then it is negative. So what is the value proposition here? This test does not require any preparatory step, equipment, as well as expertise. And it is very rapid. Within 12 minutes, you can complete the assay and color will be developed. Whereas, if you try to do smear microscopy, around 4 hours you will take. And the number of samples also, you will not be able to handle more. And for culture, you will need actually more than 1.5 months to 2 months. And for nucleic acid test also, you will require around 4 hours. It can detect low concentration of MTB also. And this method is useful in both pediatric as well as adult patients. This is not possible by other methodology. And color detection is basically then becoming confirmatory and point of care method. Of course, I am coming to that point, and where we are facing hurdle that I will describe. So today we are in a certain position that we have to think that whether we will move forward or not. Now, so if we talk about more or about this assay, is that it could identify more positives than any other methods. No other method available for detecting severity of the disease. So by other methodology like culture, NA or microscopy, basically you will be able to say that whether TB is there or not, yes or no. But the weakness of our methodology is definitely it cannot detect resistant mycobacteria, as well as it cannot use any other clinical samples for detection. So it cannot detect extrapulmonary TB. So after receiving the grant from BIRAC, we went for a, basically our intention was to go for a POC, proof of concept, because without that, by a third party, nobody is going to believe your test going ahead. So it is a long journey. And in that journey, at different time points, you have to show the results by giving the samples to others, third parties' hand, and how it is performing there. Because finally, it will work in people's hand, those who are not aware of that technology or assay idea. So we carried out the testing, the proof of concept by keeping two objectives. One is that how, before that, I should say that for any diagnostic kit, novel or new, whatever you say, you need to prove that the sensitivity and specificity values with a gold standard. Now, what we did is that we carried out culture, nucleic acid test, as well as microscopy along with our test for all these samples. And almost 300 samples were used in this study. And you can see that sensitivity and specificity values are very poor, we will say, because they are not coming to that range where they normally should be around 95% or more, or more than 90%. So we have not got that. So this is actually a setback in the sense people are more habituated to accept this kind of values, but they are not comfortable if the values are low. So second point, what we tried to address is that the whether this color then is coming from any other bacteria who can produce that biomarker, metabolite biomarker. So Streptococcus pneumoniae, we found in 11 samples. So if we are finding in 11 samples, out of that, our test was positive in 4 cases, and our test was negative. That means that biomarker is present only in 4 cases. So that means in 7 samples where this Streptococcus pneumoniae was present, but it was the biomarker was not in detectable range. So out of 300, if we find that only 4 is positive, then can we say that this Streptococcus pneumoniae is responsible for the development of that color? Probably no. So in that way, we are not finding that other bacteria are responsible. So then who is responsible? Whether mycobacteria or other bacteria? From in vitro studies, I can say that we have seen that this Mycobacteria tuberculosis is able to produce that biomarker, metabolite biomarker, very efficiently. But we need to transform that confidence from in vitro studies to the clinical samples. Whether we are able to do that or not. From our conventional studies, we couldn't prove that this is possible, that this specificity and sensitivity values are good enough to believe that our test is detecting this bacteria. But one point here I want to mention is that we said that other methodologies, all these methodologies are able to detect only actively growing bacteria. And our methodology is very efficient in detecting non-replicating bacteria. So we have to probably compare with the tests which is at least available to the best of our knowledge, which is available, that should be used. Maybe along with this also, or without this test also. So the major concerns, what we faced after this is the specificity and sensitive values are not coming up as proper. And these concerns are not from our side. It is coming up from the so-called tuberculosis experts. Presence of other bacteria who also have similar capability to produce that biomarker. At least 3, 4, or 10 samples are showing that, but we are not getting totally nil or zero number of samples of that bacteria and totally clean. But so far my knowledge says that in clinical samples you will not get that kind of data, that either or. So statistically, I believe that it is convincing that other bacteria is not responsible for the development of this color. So if the sensitivity, specificity is not resolved, it is becoming very difficult for us to get funding. And to go for this, resolve these issues, we need more experiment to be done. Otherwise, we will not be able to get this regulatory approvals, because that is not very simple in India. Here, you have to go to some authorities whom our regulatory institutions believe that if they certify, then only your product will be approved. So like ICMR or that kind of organization. So what is happening is that even if we want to get it done from them, then also we need money. And we have already spent for 3-4 years, and we are not finally able to get the fund. And we try to continue the work and collect data by spending money from our pocket, but we found that it is becoming endless. So in which stage we are now? So as I said, that a lot of work needs to be done to bring the product to the market. And we have decided to close the company at this moment because it is incurring more cost to develop the product for us, which we are not able to bear. And we are also now thinking of canceling the sub-licensing agreement with EDC and CL also as a company, and then pay all the dues by selling out our assets of the company. Anyway, meanwhile, we got one more project also from RNTCP, but the funds were not released. So what I can say is that our lessons from this whole journey is going to enrich us in future if we want to go for another similar kind of ventures. So innovation and patent should have tight binding between them and with the market also. The meaning of this statement is that, so your innovation is basically what you made that product, what it can do. And patent is basically the right you are getting it from authorities to use as your monopoly. Now, if in the patent filing, if you, number one, if you change the or if you reduce the number of claims, then basically the patent is becoming weak. If with time your number of filings fail to be granted, then also your patent rights overall, it is becoming weak. And third point is that your innovation actually is becoming weak in the market to stand on its own feet. And third point related to patent is that if you don't get the rights to the major markets, then also you are losing. So before going into this kind of venture, you should properly think that whether you are selecting the right technology and you are backed up by proper patent rights, so that you can see that, yes, it has a good future. Second is that agreements should not be done to control the entrepreneur. I am saying here, entrepreneur is that who is leading presently the innovative product and taking it forward. So at different stages, probably different people will take it. So at that time, that person is the entrepreneur. Maybe at this moment, I am doing as the job as entrepreneur. Maybe tomorrow, this product will go to a marketeer. So that person will, what I am in my words, in a very simple and straightforward and very shortcut, I am saying terming them as entrepreneur at that level. So maybe their area is little bit different, but they should get full control on the product. Otherwise, it becomes very difficult. So, in other ways, that actually I can give some example. Probably that will clarify this statement better. When we were talking about that with a marketing person, that the product can be taken to the market by that guy. So what will be the actually understanding between the marketing person and us? So that person said that we have to go for an agreement, definitely, that we have to do. And I shall not be able to pay a single rupee to you to take this product to the market. And along with that, he also said that if that product fails to go to the market, then all the expenses I have incurred, that you have to pay back from me. So this was version from one person. I talked to 3-4 persons, and more or less that similar kind of persons. So what I am saying is that I am talking about a prototype that already has been made, and some data has been generated. Now, how you are going to interpret the data, that is a choice. But if that person says that we will be able to get it through from the regulatory authority, that is our headache. But this is the condition. Now the question is that whenever it is in your hand and you are looking for a partner, most of the time probably you have to think that your product should reach the market, and the person who is taking it forward, that person should get the maximum advantage if it is becoming successful. So in that way, I thought that that is basically the concept which I didn't know when I made the company. Platforms and collaborations should be used from those who are market leaders in that particular area. So platforms are basically the incubators or those kind of places, and collaborators are basically those who are going to help you in different stages, like proof of concept, then product design, and many aspects, that data analysis, then collection of data, large scale, small scale, bringing the funds from different sources, all this. So they should be market leaders. It should not happen that you are using basically an engineering platform for purely a very intricate biology problem related product development. So probably they will not understand. Or in other ways, I can say this through this kind of example, that you want to play football, and we have taken you to a pond and showing that pond that here you play football. So it will be something like that. So that is not your right ground to develop the product. They will never understand your product. They will not be able to help you also. It is not actually a complaint or it is just an experience. What will happen? You will face problem in every aspect. Probably you will not be able to use clinical samples in your laboratory where you are working. You will, your data collection will be delayed. You will not be able to meet proper experts to make collaborator or to be associated with them to generate the data. So there is possibility that you will be misguided. And finally, you will see the funding situation is becoming very, very tight. Whether it is governmental or non-governmental, it doesn't matter, because you are not actually getting the right platform for flying. Our regulatory requirements and your initiatives. So that's why what I am saying here is that your regulatory requirement and your initiative should align properly so that you can get it through nicely. For example, for purely innovative product, we don't have our regulatory structure properly placed. It is well-known fact. And that's why probably still just few days back, our central minister said that we are currently importing 85% of the diagnostic related products. That should not continue. But if we want to develop a Me Too type product, then what happens is that the Me Too type of product, if it is Me Too type of an imported version, then probably you will get some advantage. And our regulatory system is very much good, very good in that. I will say that they are very good in taking the regulatory process nicely to get it done, that approval done. But if it is a novel product, then it becomes very difficult. Then they will say that you go to ICMR, you go to that place, that other place, and get it done, and then you give us that certification. Now, what happens is that where we will go and how much money will be required for that. We found that it is very difficult. So that's why probably you need a combination of third-party evaluation, investment requirement, or marketing partner they all need basically a accelerator support. What is accelerator? Accelerator is basically the domain experts, and they are ready to help the innovator and the startup company to identify, they are domain experts, so they will identify your product, they will evaluate your product, they will then help in every stages that you should do in this way, then what data you are getting, then and how that data should be presented, pitch, making that pitch deck, and then they will bring the investor also. So there are different kinds of investors, small to big. So based on the requirement and all those things, the area, which investor will be interested, they know all those things. So they will bring the right person and feed the technology with proper funding to support particularly the product to come to the market level. So that's why what I found that, and this team is going to be the most important part of the company. So the company should be made with marketing guys. Maybe you can induct the marketing guys little bit later, but not too late. Regulatory guys and subject matter expert. So if they are there, then they will be able to help you to get the right people. I am talking about this kind of team, even having accelerator support also, because you need the right frequency to understand what accelerator is saying to your company. So with this, I want to stop here. I hope that I have said the major points related to the company during my journey. Whatever I have understood or learned, I try to share with you guys. So if you have any question, please.
Yes, sir. So thanks for sharing your journey with all of us. And definitely you have highlighted what are the takeaways or what one should do or one should not. So there have been some questions over here. So what is the spin-off? I mean, how it is different from, I mean, startup and spin-off, are they same?
Basically they are same, but spin-off is basically a startup. As an individual, I have made a company, startup company, okay? But when I am an employee and I have developed a technology in NCL, CSIR, and CSIR is allowing me to take the technology out of NCL, and they are giving, with certain conditions, a freedom to develop the technology and bring it to the market. So then it is called spin-off.
That means, I mean, the institution with whom you are working, you develop it there and then you take it away.
Yes, yes. Now, actually, in that aspect, I can say that now I will say that if I am given now a technology, then I will take it after regulatory approval.
Right.
I will not take at the very early stage. Or otherwise, I will look for a clear assurance that CSIR is going to provide me all financial and technical support to take the product to the market.
Right. Sir, there was one more about the technical part of it. Why early morning sputum was required? I mean,
Early morning sputum is required probably because that sputum is there collected during the sleeping hours, and in the early morning you are coughing up the sputum. So it will, in all probability, will contain that biomarker definitely, and our result also has proven that we surely get that.
but then it might work in any time, timing of the day.
Depends on the availability of that bacilli. If the bacilli number is low, say in the lower side, then what will happen is that during your sleeping hours, 6 hours, you have not cough of anything, okay, and it has been collected. And when you are in daytime, you are moving here and there, and repeatedly you are coughing up and throwing it. So it will not stay there because you are doing that same job repeatedly. So it is getting empty on a regular basis.
Yes. So there is one more question. Is the test resistant to temperature? I mean,
temperature, place, all those things has no problem. So you don't have to worry about that.
right. Sir, about the startup, there is a question. Can you suggest the techniques or approaches by which we can cultivate the innovative mindset in teachers as well as the students? I mean, the way you have thought about your research, how people can think about their own research? I mean, what are the things they should do to cultivate this kind of mindset?
Yeah, actually, what has happened, during during my PhD thesis also, I worked on different problems, and first of all, what you have to think is that in which area you will be working or where the problem lies. Actually, when I started working on tuberculosis at NCL, before that, almost I have spent more than 8 to 9 years during my, you can see from my CV that I have already spent 8 to 10 years, yes, more than 10 years. And I never used MTB. I have not worked on MTB. And when I joined NCL, then my one thinking that worked was that what is latent TB, what is non-replicating TB, why people don't work on that. If replicating bacteria, they can be killed, and if non-replicating bacteria is not killed by the drugs, then probably that non-replicating bacteria is the problem. So then I started looking into that, and my whole research is on that. And we have become very successful in that part. And always I thought that whatever new idea or new technology we are working on, new information we are getting, how we can capitalize it in terms of patent filing and whether they can be converted into a new product or not. So in that way, we have always thought. So that is the kind of mindset I have developed. Yeah.
Yes, so you have 35 applications to your credit. So definitely. So there is one more question about funding agencies for pharma startup. I mean, are there any agencies who are specifically funding the pharma startups?
I guess that I don't have any knowledge about that. That only pharma they are giving. Actually, frankly speaking, the funding itself is a very rare thing in India. Right now, whatever you are getting, this is another aspect I didn't actually cover. That is that your entry into this whole ballgame, that can happen very smoothly. Say you can start a company, you will get probably a funding from this BIDAC or DBT or whatever may be. And after that, your second or third funding will become very crucial. And that is basically called as death valley. So where we have also started.
There is a question on private investors. I mean, approaching private investors. How do we do that?
Actually, we try to contact. Actually, we through our different sources, we talk to private investors. Actually, private investors doesn't have a proper, well-trained mindset how to deal with the things. What they think that this is suitable for them, then they will invest. Otherwise, they will not invest. So you cannot expect a very routine kind of character from them. Rather than that, I will say that the investor, all the private investors, but network investor or they are regular investors, they will be much better.
So you mean to say rather than approaching a government funding agency, it may be better to approach a private investor?
No, private investor will be better in the second stage or third stage.
In the second stage.
Yes, yes. And obviously the challenge is there from your first tranche of funding from government, obviously you have got and you have started, you should complete your proof of concept and go beyond that so that you can convince the private investor nicely to get the funding onwards.
So there is one question over here. What sort of support we can get from the government R&D departments like NCL, let us say? So to start to have a new startup in R&D.
So you have a startup. You are asking that you have a startup and
no,
you don't have so many.
We want to come up with a startup. So what kind of support NCL or any government agency would be able to provide?
Yeah. Actually, I can tell you, recently one memo has come from the ministry, central ministry probably, that all these national laboratories or regional laboratory or whatever may be, they are well funded. They have lots of equipment, but these startup companies, they don't have this kind of facility. Whenever they will require this kind of support, that startups should get the support from the institutes or laboratories, that national laboratories or regional.
I mean, one who wants to have a startup has to approach the laboratories
Right.
with a proposal
Laboratory.
and
Yes.
right, sir. Thank you very much for answering all those questions and sharing your journey with us. I would request, sir, this is a token of our appreciation certificate generated for guiding the gathering of teachers. And thank you very much once again. I would request Dr. Gandhi to take over.
Thank you, Valekar sir. Thank you, Dhiman sir, for sharing your knowledge, expertise, journey right from the scrap to setting up the own enterprise, the hurdles, and still going on. Thanks a lot for that. I request participants, you can even log off and join at 2:25 for the post-lunch session from Dr. Smita Kale from Venture Center, National Chemical Laboratory. Thank you.
Thank you. Thanks.